In this video, we're going to demonstrate the participant comparison and code grouping features in version 4 of the Participant Explorer. And, on the way, we'll point out features not shown in the earlier videos. The starting page shows the new comparison option, which we'll use to explore the new features in version 4. We can compare up to 12 participants by entering their IDs as a comma-separated list. We can obtain these IDs from other applications in the research environment, or by using Participant Explorer itself, by downloading a list from a previous search. We'll be using 4 test participants that have been created using synthetic data. Note that we can only see the Compare tab when we start from the comparison entry point, and that one of the changes in version 4 is that many of the buttons to perform actions have been removed, and navigating around the app is primarily done via the tabs. We'll click the Search button, or the Result tab, to perform the search. The Result tab provides an overview to confirm our selection. From here, we can proceed to compare participants via the Compare tab, or step through them one by selecting a row in the table or by clicking on the Participant tab. A new feature in the Participant Details panel is the Family Case Solved results of the GMC exit questionnaire at a family level, together with any additional comments provided about the case. This is available for most rare disease participants. We can follow the link here to jump directly to the full entry in LabKey to get more details. Just below this, we can see some useful metadata about the genomic sequencing, if available, including the plate key and a link to the genome file path. If we copy that into a new tab in the browser, it will show the genome files in the directory in the Research Environment file system. A link in the top of this panel will again take us to the source of the data in LabKey. The date on which the sample was taken is here too, and will appear on the timeline below, as this could be useful to see in the context of events before and after it. The 4 icons on the top right of this page take us directly to the section of the page we're interested in. In this case, we'll jump to the timeline. Notice that part of the bar sticks to the top of the page to allow us to jump back or to sit on the timeline as we page forward to the next participant in our results list. Selecting the counter between the forward and back arrows takes us back to the results. Back on the timeline, we will now look at some new tools that have been created to help you filter and group the data. Although the plots for these test participants with synthetic data are quite small, some of the real medical histories can be much larger, and we'll be able to take advantage of some of the filters to focus on different parts of the data. The Include Data Categories control is a coarse-grained filter with which we can remove large blocks of data. By default, the diagnosis and procedure codes, which are coded as unknown, are excluded by being deselected, but they can easily be re-added. Adding further filters, for example, we can choose to deselect HPO terms if we want to focus on the ICD-10 codes. Toggle View by Participant Age to translate the dates of the encounters relative to the participant's age. The Code Grouping slider groups codes that are in the same coding hierarchy, for example, that share the same parent code, then grandparent code, and so on, applying grouping higher up the ancestor hierarchy as the slider is moved. This allows commonality to be found between events that have codes in related conditions or procedures that would not be common at the most specific code in the raw data. We'll look at this in more detail on the comparison page. The Show Subsumed Codes option turns on the display of how many codes are subsumed by a row in the y-axis when grouping is active. When hovering on each data point, it enables a display of the exact codes subsumed by that data point. The hierarchy information can also be used as a filter by using the Top-Level Concepts filter. Although it only filters using the highest grouping level available in the data, it is significantly more fine-grained than the Data Categories filter, and replaces and improves the previous code groups filter. The text or code filter is usually very fine-grained and will include only data points whose current display, description, or code string, according to the level of grouping and filtering, has a match. In other words, the text filter is applied after the other filtering and grouping has been done. We can use this to find whether a code or condition or partial text exists in very large charts instead of poring over many rows of data. The text filter can also be used as a broad filter when used to target the different code systems, for example by entering ICD. Using the available filters in combination should enable complex timelines which don't fit on the screen to be cut down to more manageable chunks. To view the timelines of multiple participants at once, go to the Compare tab. In this tab, you can see the medical histories of multiple participants together. So if, for instance, you had a small set of participants with the same rare variant or rare somatic mutational signature, you could use this feature to see if they had any clinical phenotypes in common. The page displays with View by Participant Age turned on by default, no code grouping, and the participant shared codes threshold set to 2. This means that the only codes shown will be those present for 2 or more of the displayed participants. As already mentioned, the code grouping control can be used to group the clinical codes on the y-axis by their common ancestors, based on the hierarchies in the classifications and ontologies. At the time of this recording, in August 2021, this includes ICD-10, OPCS, HPO, and SNOMED. Different positions of the code grouping slider correspond to different depths in the code hierarchy. 0 will display the most specific codes available, and 5 will display the highest, least specific ancestors. Please refer to the help documentation for details about the exact correspondence for each classification and ontology. To see how this works, we'll first hide all data categories except for HPO terms and conditions and observations. We'll also start by setting the shared codes threshold to 1, effectively disabling the commonality threshold so we can see all the HPO terms and ICD-10 codes that exists for any of the 4 participants. By default, code grouping is set to 0, which means no grouping is applied, and the y-axis displays the codes as they appear in the source data. This is the highest level of detail available in our dataset. If we now set the shared codes threshold to 2, we see that most of the codes are filtered out. Only 2 of the HPO terms and 2 of the ICD-10 codes are shared by at least 2 participants. So it appears that the 4 participants did not have very much phenotypic overlap. By increasing the grouping level, codes will be replaced by their parent codes, and when moving the slider further up, by the parent codes of their parent codes, and so forth, until the maximum level of grouping is reached. Because the grouping level is also the input for the shared codes threshold, we can expect to see an increase in shared codes as we increase the level of grouping. Let's set the code grouping in the middle, between 2 and 3, and see what happens. Some of the HPO terms and ICD-10 codes are now grouped under ancestor codes. The effect of this is that more codes now pass the shared codes threshold, and more data points are visible on the timeline. For example, the HPO term for chronic kidney disease and the ICD-10 code E66 obesity. At this point, it can be useful to tick the Show Subsumed Codes checkbox to see which original codes have been grouped under each ancestor code. For example, we can see that the different HPO terms for Stage 4 and Stage 5 CKD are now grouped on the same row under chronic kidney disease, and a common phenotype of participant PX300 and PX400. We can also see the HPO terms can have multiple parents. Short middle phalanx of finger and aplasia-hyperplasia of the middle phalanges of the hand both group the same 3 original codes. Similarly, ICD-10 codes E66.9, obesity unspecified, and E66.8, other obesity, are grouped under E66, obesity, and a common condition of participant PX100 and PX300. Now, let's increase the grouping to level 4. The ICD-10 code range for glomerular disease has appeared, showing codes NO5.9, NO2.8, and NO4.9 as a common condition of PX300 and PX400. We also see a lot of new HPO terms, representing various ancestor concepts for the source HPO terms at this level in the ontology. Finally, at the maximum grouping level, all codes are grouped under relatively generic concepts, such as abnormality of the genitourinary system and neoplasms. This high-level view may be useful for looking at a large number of diverse data points in a compact manner. The download page allows us to export information about the participants to the research environment desktop. Download in comparison mode differs from downloading clinical and concept search results in that only participant-level details can be downloaded. Condition and procedure codes are not available for download. For more details on group coding and all other features of the Participant Explorer, please visit the Help pages via the menu in the top right. Thank you for watching this video.